Okay, in this video we need to take a look at mitochondrial transport. Now there are two different types of molecules that need to be transported into mitochondria. We have the small molecules that are involved in metabolism, and we also have proteins. Now the protein transport mechanism is best left to a cell biology course because that's going to require some information about chaperones and membrane fusion complexes, and that is beyond the scope of this particular course. So we're going to look at the small molecules that are transported in and out of the mitochondria. We have to balance our small molecules that are moved in and out of the mitochondria in order to make sure that we have sufficient substrates for energy production. So remember that the inner mitochondrial membrane is the membrane structure that is very regulated for transport. The outer mitochondrial membrane has a porosity to it. There's pore proteins in there that allow proteins and small molecules that are less than 6,000 molecular weight pass. So typically when we're talking about small molecule transport, we're not considering what is going on with the outer mitochondrial membrane because free diffusion moves very quickly through that membrane, through the pores. So what we have to consider here are the components that are necessary inside the matrix of the mitochondria for the subsequent chemical reactions to generate energy. Now the first one we've examined before, and this is the movement of the final product of glycolysis into the mitochondria. Recall this is the molecule pyruvate. Pyruvate is transported by the pyruvate translocase. This translocase is a transmembrane protein that crosses the inner mitochondrial membrane and allows passage of pyruvate into the matrix. Now to drive this process, we are going to take advantage of the proton gradient that is established by the electron transport chain. So recall that complex 1 is going to pump protons into the intermembrane space. Complex 3 also does the same function, and finally complex 4 also is involved in pumping protons. This is generating our proton motive force that we saw in a previous video, and it is also considered an electrochemical gradient. An electrochemical gradient by the fact that it's an electrical gradient because we have sufficient positive charge on the outside of the inner mitochondrial membrane because the protons are positively charged and we have a relatively high concentration of them out here. And that, of course, because you're moving charge, that's going to cause a charge displacement. And so the inside of the inner mitochondrial membrane will be slightly negative relative to the outside. So moving pyruvate into the mitochondrial matrix, we are going to take advantage of this gradient. And we are going to use the gradient to transport pyruvate. So recall this is secondary active transport. This is also a symporter because we are moving two molecules in the same direction. Now the other molecule that we have to be able to get into the mitochondria, of course, is ADP. Because recall, we have our ATP synthase enzyme complex with our head molecular motor, and the job of this is to take ADP plus inorganic phosphate and generate ATP. So the question is where does the ADP come from? Well, recall that energy is primarily used in the cytoplasm. And in the cytoplasm, ATP is being hydrolyzed to ADP plus inorganic phosphate, and this energy that is being released, being an exothermic reaction, is driving many cytosolic functions. The most obvious example we've seen is skeletal muscle. The hydrolysis of ATP is required for the actin-myosin ratcheting function for contraction. So the ADP is in the cytoplasm. We have to get the ADP into the matrix of the mitochondria. So the way we're going to do that is we are going to couple ADP movement inside the matrix to the release of ATP. Now this is a one-to-one transporter. It is also called an antiporter because we are moving two substrates in the opposite direction. And being a one-to-one transporter, it works out great stoichiometry-wise for the function and product of the ATP synthase enzyme. So as ATP is being synthesized, it is having binding to the inside and ADP bonds to the outside, and we get a translocation event. Now the other thing that drives this process is recall that ATP has 4 negative charges. ADP has 3 negative charges. Recall that the gradient that we have of protons is an electrochemical gradient, and so that the imbalance of charge between the ATP and ADP, the extra negative charge in ATP, is being attracted to the positive charge on the outer surface of the inner mitochondrial membrane. That is also assisting in the movement and transport of ADP and ATP. Now finally, we haven't talked about inorganic phosphate here. We have to be able to import inorganic phosphate, and so we have a transporter here that is a symporter. And as you can imagine, a symporter means that there's 2 ligands, 2 substrates that will be transported in the same direction. We need inorganic phosphate inside the cell, inside the mitochondrial matrix, and so we're going to take that from the cytoplasm. Again, we have a excellent source of inorganic phosphate because of the hydrolysis of ATP, so that's coming from the cytosol. And as before we've seen with the pyruvate translocase, we are going to take advantage of the proton gradient. So this symporter will bind inorganic phosphate and protons on one surface and transport those together to the matrix of the mitochondria.